Hello, can you see the slides? Hello?
Yes, Doc.
Can you see the slides in the right way?
Yes, Doc.
Yes,
Yeah,
Doc.
okay, I was trying to define pesticide resistance. So how does resistance occur? This resistant biotype survives the repeated pesticide exposure and reproduces. So those pests which have survived the pesticide, they will start to build the population. So the pest organisms population may become dominated by the resistant biotype. At this point, the pesticide is no longer useful, and other management options must be used if they exist. If resistance is managed effectively, the pesticide remain useful to growers. So this could be a demonstration of pesticide resistance. Probably the red larva probably represent something which is resistant, and the green ones may be resistant. They're going to be, they're going to be killed. They're going to be killed by the pesticide. These are susceptible. Now let us look at the effect of repeated use of a pesticide. If we keep on using the same pesticide over and over again, so using one type of pesticide over and over again can produce resistance in a population of either insects, fungi, weeds, or any other organism. The offspring have the same genetic composition as their parents and are able to withstand the pesticide. The pesticide is no longer usable as a viable method of pest management for this organism, since its surviving biotype is now resistant to the pesticide. If we look at the insect resistance, insecticides are used to control insects. Insect resistance to the synthetic insecticide DDT was documented as far back as 1947. Since that time, within 2 to 20 years of the release of new insecticides, key pests have actually developed the resistance. This causes a more frequent application of the insecticides. Growers must ultimately switch the insecticides as insect control diminishes. Once your pesticide is no longer controlling the target pest, it's better to switch to another insecticide or pesticide. Even in plant insecticides such as BT can be overcome by insects in due course. Now let us address this issue. How insects become resistant? What causes them to be resistant? There are 4 points on this issue. The first point is based on metabolic, and I have several examples of metabolic resistance, or resistance which is due to metabolism. The insect can clear its body of toxin or break in toxin down quicker than other insects. So in an insect pest where the processes are fast, it can survive. Now there's what we call a target site, where a pesticide can combine with the target site. If there's failure to combine at the target site, then the pesticide is not going to work. It's a form of resistance. So the insect can no longer connect at its target site at a molecular level in the insect. And as a result, the insect will survive. Then there is the penetration. I have just talked about the cuticle, its role. It can prevent the pesticide entering the target organism. So the insect's shell more slowly absorbs an insecticide, and therefore the insect becomes, it will survive. But this can differ across insects. So the cuticle of the insect can actually prevent the penetration of insecticides, and this can actually cause resistance. The other issue is behavioral. There could be certain insects which can actually be killed by the insecticides, but because they can sense this insecticide, they will avoid the places which have been sprayed. So by avoidance, they escape the impact of the pesticide. But if they come into contact with the insecticide or pesticide, these insects will die. So if I take this metabolic issue further, I'm no longer going to talk about the target site penetration. I've talked about the cuticle, and you should also remember this behavioral issue. We have malathion resistance. The resistance to malathion is caused by raised levels of carboxylestrase activity and resistance in aphids. You've got aphids with this enzyme, they can destroy malathion, and as a result, they are not killed by malathion. I think that is the important issue to remember. These enzymes are capable of hydrolyzing ester linkages of the ROP groups, the organophosphates. So the aphids can survive because of carboxylestrase enzyme activity. Methylparathione can also be hydrolyzed by a resistant strain of potato aphids, also using the same enzyme carboxyl esterase, so it can protect the aphids. Now there's this pesticide tetrachlorovinphos, which is resisted by houseflies. Now the houseflies are able to dealkylate, they are able to remove certain elements of this pesticide, and renders it non-toxic. And these houseflies which are resistant, they can actually break this insecticide very fast. The biochemical reactions are 120 times, probably in terms of being fast. They are 120 times more than the susceptible strains. So the speed at which the breakdown occurs is 120 times, which means these houseflies will survive. And the glutathione S-transferase system could be responsible for the resistance in the resistant strains. So remember the tetrachlorovinphos resistance in houseflies, which is aided by glutathione S-transferase. We also have the carbamate resistance. The resistance to carbamates is predominantly due to gene selection, leading to more microsomal oxidation, but it is mainly the activity of cytochrome P450 in resistant insects that cause the degradation of the carbamates compared to susceptible pests. So we have got the activity of cytochrome P450 in breaking down the carbamates. Probably, if you remember the phase 1 activities. Organochlorine resistance, this is aided by the enzyme dehydrochlorinase, which is present in the resistant pests or strains. So this enzyme will cause fast breakdown in resistant strains compared to susceptible strains. Organochlorine resistance due to the presence of dehydrochlorinase in some pests. So that was it on the metabolic processes which can aid resistance. But there are other, processes which can lead to the resistance of these pesticides. Now, you may be interested to note that in this country, we have developed management practices to contain pesticide resistance in insects, especially in cotton, where we have the cotton bollworms, The Eriothis amigera, I think it's now called the Helicobacter amigera. You may have to check on that. Probably Eriothis is an old name, which was replaced by Helicobacter amigera. And also the mites, the Tetranicus species. These two have a potential to resist insecticides. The mites, the Tetranicus species, they are controlled by acaricide rotation scheme, while the bollworm, the pink bollworm, was managed by the closed season. I will explain these issues. So in order to manage insect pest resistance in cotton growing areas, number one, we've got bollworm insecticides, which which are alternated during the growing season. So when you put pesticides, these are alternated or rotated during one season. As for the acaricide rotation scheme, it is also intended to reduce the development of resistance in Tetranicus species, the mites. So we want to reduce the resistance in mites. So we have to rotate the acaricides. Then we have a situation which we call the closed season. This is the time when we instruct farmers to remove the cotton plants. Actually, there's a legislation which forces farmers to remove their cotton in the fields during certain times. This is done in order to break the cycle. I think it's for the pink bollworm. Now, we can use this table probably to explain the closed season, what we call the closed season. In southeast low veld and the Bight Bridge area, all the cotton should be slashed by 1 August and destroyed between 1 August and 15 August. So the cotton plants should be destroyed and the planting should commence around 5 October. So this is done in order to manage, I think, the pink bollworm in the southeast low veld and the Bight Bridge area. Now, as for the remainder of the country, from 15 August to 10 September, all the living cotton plants should be slashed as from 15 August and destroyed by 10 September. As from 20 October, cotton could be planted. So this is done in order to break the life cycle of the bollworm. I think it's pink bollworm. So that is it on the closed season. Just remember that there are certain dates when cotton is not allowed to be in the field in order to break the life cycle of the pink bollworms. The other management technique in order to break resistance is the rotation of pesticides in cotton. The idea is to slow the development of insecticide resistance of bollworm insecticides, and these have to be rotated. We have synthetic pyrethroids which are used in the rotation with the conventional insecticides, mainly carbamate group, I think also organochlorines. The rotation of bollworm pesticides in Zimbabwe is known as the pyrethroid window. So we should know where to locate this pyrethroid window. This window was developed by the Cotton Research Institute, Department of Research and Specialist Services. Here are the details when we are rotating the pesticides in cotton. Pyrethroids must not be used in Zimbabwe before 25 December or after 1st March in southeast low veldt. Use of pyrethroids is confined to the period from 1st February to the end of growing season within non-southeast low veldt areas or remainder of the country. Before and after these periods, we can use conventional pesticides. Probably to make this clear, we may have to use this table which divide the country into two regions. We have got the middle veldt, beginning of season to 1st February. We can apply conventional insecticides. These are the carbamates and the organochlorines. Then from 1st February to the end of the season, we can switch to the pyrethroids. That is in the middle veldt. In the southeast low veldt, from the beginning of the season to 24 December, we are using ordinary insecticides, either carbamates or organochlorines. Then from 25 December to 28 February, it's a narrow window where we can come in with the pyrethroids. Then beginning of March to end of season, we switch back to ordinary insecticides, the carbamates and the organochlorines. So in the middle field, it's quite straightforward. But in the southeast low veld, it's a bit complicated because there's a narrow window where we switch to the pyrethroids. These are the pyrethroids: lambda, silo 5 EC, fenvalerate 20 EC, provavinet 2 EC. I think this is delta methrin 2.5 EC. Conventional bollworm chemicals, you can have a Cabril 25 85 WP. You now know what these mean. Thionex endosulfan 35 EC. So here we've got the details of the pesticides. So we have got examples of organochlorine and carbamate pesticides, which are rotated with the pyrethroids, which are also indicated in that table. Now we move on to the system for red spider mite. Dimethoate was recommended for the control of red spider mite as far back as 1986. Actually, the resistance of red spider mite dimethoate was recommended for the control of red spider mite. This was reported by Brettel, a cotton scientist working at Kadoma Research Station in 1986. But the resistance of red spider mite was noted in 1968 when dimethoate was being used. And it is closely related to organophosphates such as demetone, S-methyl, and thiometone. So what causes this resistance in red spider mite? It's overexposure of the red spider mite to chemical stress. There's also a short generation turnover time, which is coupled with fecundity. This is how reproduction occurs in this red spider mite. So you may note that the acaricide rotation scheme was recommended as far back as 1973-74 season. That is when it was introduced from the report of Dan Kobe, 1973. According to this acaricide rotation scheme, Zimbabwe was divided into 3 regions. So you had registered systemic or translaminar acaricides, which are classified into 3 distinct chemical groups, among which cross resistance is not likely to occur. Yeah, it is very important to note these acaricides. So Zimbabwe was divided into these three regions. In their recommendation, they said that in any region, each group of the acaricide can be used for 2 consecutive seasons without making some changes. After that, you can switch to another group for again another 2 consecutive seasons. So it is important to remember that Zimbabwe was divided into 3 regions, and in each region you can use a certain type of insecticide, acaricide, for 2 consecutive seasons before you switch to the other type. That is very critical. So here are some of the recommendations from Joa and Mvuta in 1993, where they are giving recommendations for those 3 regions. But what is critical is to note that each acaricide can be used for 2 consecutive seasons in each area before switching to the other. So there may not be any need to memorize this table. So the red spider mite, the chemical or pesticides which are recommended for its control in the acaricide rotation scheme are amitraz, triazophos, provenofos, tetradiphon, and clofeniapa. Very complicated names. So this is it on insecticide resistance. Now, as far as fungicide resistance are concerned, the fungicide resistant strains are already in the population and caused by natural mutations. The resistant population will build up. Once the population of the fungicide resistant mutants is high, then there's the loss of fungicide efficacy. The fungicide will no longer control the disease. What are the causes of fungicide resistance? The fungi could possess alternative metabolic pathways to one targeted by the fungicide. Therefore, it may survive. The ability of the fungi to degrade and inactivate the active ingredient in the fungicide, just like what other insect pests do to insecticides. So how do we avoid fungicide resistance? Use of recommended rates, avoid unnecessary sprays, rotate different types of fungicides, use alternative control methods, or we can use integrated control methods. So for the fungicides, this is it. But we had a report from Tasa et al. In 2015 from a study which was done under a greenhouse on tomato, cucumber, what they reported was that Phytophthora infestans was resistant to most of the fungicides, with the exception of diclofenac, dimethyl, ammonium chloride, which controlled these pathogens. Fusarium oxysporium also exhibited some level of resistance. So there was a bit of resistance which was reported from this study. We move on to weed resistance to herbicides. Before we go any further, I would like to say that in Zimbabwe, we have not yet recorded resistance under herbicide control programs. But of course, it is important for us to have some knowledge about herbicide resistance in case it may develop. But so far we don't have any reports about herbicide resistance, or we haven't looked enough for it. These are the reports of herbicide resistance. More reports are increasing. Now the blue areas on the world map are showing where herbicide resistance is a problem. Australia, South Africa, I think Australia and North America there, it's a problem, and in Europe, then there's the lighter blue, some parts of South America, South Africa, and some parts of China. New Zealand, we have some problems there. But if you look at Zimbabwe and other countries, I think Egypt, Sudan, and probably Kenya, they have a bit of resistance there. But in Zimbabwe, I don't think we have problems. And these are the white areas in different countries, There's no resistance. Here I've got a table. This is from the US, where they recorded herbicide-resistant weeds in Iowa State. Maybe you may have a closer look later. These are not the weeds which we are having. We probably could have some of the weeds, but in this country we do not have resistance. So when susceptible weeds, which are normally killed after the application of the herbicide at the recommended rates, survive, it means these weeds are resistant to the herbicide. And under field conditions, we have not yet experienced this. What we see are probably calibration problems leading to poor weed control. So this is how resistance can occur. It can start being very low with the time. As the weeds which are resistant increase, then resistance also increases. So here we want to see how resistance can develop. We have got the first box to our left, representing a field which was sprayed with herbicides, and probably there's that green plant which could be the resistant weed. With the time that it was ignored, or normally we ignore, it's only one plant. There's no big deal sometimes, to kill every weed. So that weed may produce a flower, then it produces seeds. If you follow the arrow down there to our left, we now have a bit of plants there, probably 3 plants. Maybe we ignore them, they produce more seeds, and eventually we are confronted with the majority of species which are resistant. And at that stage, when we have all the weeds being green, as represented by that color, the herbicide is no longer useful. And there we say we have a resistance. But such a situation is never developed in this part of the world. But in South Africa, yes. Again, we talk of weed resistance, then selection pressure. If we subject the weeds to the spray of the herbicide, the selection pressure and that red plant will remain and it will multiply. Then it produces a resistant population, which will cause poor weed control in due course. So if we focus on the weed population, so in a population of weeds, the frequency of weeds with resistant genes represent a very small proportion of the total population. What we are saying is that within a weed population, there are always genes for resistance, but at a very low frequency. So the next issue is that the frequent use of the herbicide will create selection pressure on the population of weeds. The population of resistant weeds will now increase and become the dominant weeds. Now at this stage, when you have a dominant population of resistant weeds, the herbicide will fail to control these weeds. And we say we now have herbicide resistance. These are some of the weeds which have been reported to be resistant to herbicides. Chenopodium album, Avena fatua. I know some of these weeds we have them, but we haven't registered resistance. Elusini indica, we have Elusini indica here. Amaranthus hybridas, we also have it here, but ours is killed by the herbicides. But in the US, some of these weeds have now developed resistance. So let us look at the mechanisms of resistance in order to understand how weeds become resistant. Probably we may need to revisit what happens to These herbicides, if they enter a plant system, we know they are broken down. So it is this process of breaking down the herbicides which can lead to resistance. So mechanisms of resistance, we have got the target site resistance, where we have the enzymes, a change in the primary structure of the enzyme. We have a single point mutation on the gene. Then at that site, the herbicide will fail to combine with the enzymes. If I can use a diagram to illustrate this, so at the target site, we have got the herbicide represented by the triangle down there, and that wedge where it's going to fit. If it can fit, then there's plant death. If there's alteration, then it cannot fit, then the plant will survive. Here we are trying to describe the molecules which are present at the target site, which are supposed to combine with the herbicide. These molecules are actually enzymes which should combine with the herbicide. And if there is failure to combine, then there is resistance. If we look at acetyl coenzyme A carboxylase ACCase, now acetyl A carboxylase gene has been reported in grasses because it has been altered in avinifaciol. Some of the herbicides which kill grasses will fail to kill avinifaciol. What I refer to as FOPs and DIMs, these are the groups of herbicides such as allyloxy, phenoxy, carboxylic acids, cycloheximdiones. You can see that's why we say FOPs and DIMs, just to shorten the complicated names. Acetolactate synthase. We have sulfonylurea and imidazolinones. If something happens at that enzyme, then all these herbicides will fail to kill the weeds. Mutation can also happen at the D1 protein. Then we have triazine resistance. This has actually been registered in weeds. Photosystem 1 diverting herbicide. We have detoxifying oxygen species. Now, some weeds which are supposed to be killed by a lot of oxygen which is released in the form of superoxide dismutase and ascorbate reductase, glutathione reductase. These are actually enzymes which deal with the oxygen production. You may have Coniza bonariensis or Coniza species which can immobilize paraquat. And that Coniza is able to detoxify the oxygen species. It's very rare to find, plants in Zimbabwe which are not killed by paraquat. But in other countries, they now have, plants which paraquat fails to control, which is quite surprising. Then we've got resistance to cell division inhibitors, use of dinitroanilines, e.g. trifluorine, which normally kills these grasses, but there's now resistance being reported, for example, in Erosine indica, Setaria viridis, and Sorghum haripens, which are all grasses, which are supposed to be killed by trifluorine. Resistance to glyphosate. There are few reports of weed resistance. This is also surprising because we find glyphosate killing almost any plant in Zimbabwe. But guess what? In Malaysia, Erosine indica, sometimes you can fail to control Erosine indica using glyphosate, which is quite surprising. This one inhibits the ESP synthase. There's also twice transcription of EPSP synthase, probably in Erosine indica. But we know if you apply glyphosate to Erosine indica in Zimbabwe, that Erosine indica will be killed. But in countries like Malaysia, Erosine indica now survives. These are now certain types of Erosine indica with the genes to resist glyphosate. Also, resistance to auxin type herbicides, things like MCPA and 2,4-D. We have macroprop to chickweed, which is resistant to this herbicide. Herbicide immobile in resistant weeds. For example, like what was reported in New Zealand, where you have MCPA and 2,4-D failing to kill weeds. We also have something which is known as enhanced metabolism resistance, where you have cytochrome P450s, we know what they do, and the glutathione S-transferase. These enzymes have been implicated in weeds, so their presence may cause a breakdown of herbicides. This is just a model to explain what can actually happen. In the first diagram, where we have the herbicide being represented by that triangle, if it closely fits to the TS, the plant will die. But because of the metabolism, you have your herbicide triangle which breaks, represented by the arrows there. It's not going to fit there. Or alternatively, it can combine with something, then it becomes a large triangle. This is conjugation. Then it cannot fit, and this can lead to resistance. We can also have enhanced activity of P450, which has been reported in some weeds. Also, the role of GSTs in metabolism of atrazine, aracro, and metaracro. These can actually detoxify the herbicides. If it happens very fast, it means the weed won't be killed. Another issue called enhanced compartmentalism or restriction of the herbicide, either in the vacuoles where it is not going to reach the site of action. We have something called the ATP binding cassette, ABC transporter, which moves xenobiotic substances or herbicides from cytoplasm cell vacuole. I think it's from the cytoplasm to the cell vacuole. We can have conjugation to glutathione also taking place. So if the herbicide is quickly moved to the vacuole and kept there, then it's not going to damage the plant. So we have got the first model there where you have the herbicide fitting to the, it could be to the enzyme and it causes death. But if you look at the diagram below, the herbicide is moved straight into the vacuole. It has no chance of uniting with the plant to produce something which is deadly. I just want you to note two things here. There is something which we call cross resistance. It's a resistance to a number of herbicides due to only one resistance mechanism. If there's only one resistance mechanism to a number of herbicides, that is cross resistance or altered target site resistance to many herbicides. There's also something which we call multiple resistance. This is resistance to two or more herbicides due to the presence of more than one resistance mechanism. For example, like ACCKs and ALS inhibitors. So with the first one, it's only one target. The second one, multiple resistance, many targets. So what are the factors that can speed up resistance or that can cause resistance to develop? Number one, it's the initial frequency of resistant individuals in the population, their proportion, size of seed population in the seed bank, and the length of time seeds remain viable, especially of the resistant types. The susceptibility of weeds, then the repeated use of herbicide, single mode of action here. If we keep on using the same herbicide over and over again, eventually we may build up resistance. Then the use of herbicides with a long period of activity, this can create problems of resistance. Use of herbicides with a highly specific single mode of action, like some of the new herbicides. This can lead to resistance, especially these sulfonylurea herbicides, which are very which are very specific. Now, strategies for managing and preventing herbicide-resistant weeds. We can rotate herbicides with a different mode of action. This can actually reduce resistance. We can use herbicide mixtures, herbicide with a different mode of action. We can use herbicides when only necessary. Control weed escapes, those weeds which escape, and the sanitation of equipment to prevent the spread of resistant weeds. Use herbicides with short residual life. This will reduce the selection pressure. Scout the fields for resistant weeds. Need to remove weeds which are not controlled by the chemical. Also adopt integrated weed control practices. We shall talk about these integrated weed control practices later. Adopt crop rotation. Crop rotation means using herbicide program, making it difficult for resistant weeds to increase. In a crop rotation, there's the opportunity of using different herbicides. So probably you can reduce this risk. But what is important for us, what I was talking about is theory in terms of the Zimbabwean situation. But we need that theory in case we have the problem developing in this country. So probably what could be interesting to us is why there is no herbicide resistance in Zimbabwe, which is a good thing. Maybe if we know why we don't have it, we may maintain this situation as long as possible. Number one of the reasons why we may not have it is that herbicide resistance, actually herbicide resistance has not yet been reported in Zimbabwe. Whether it's there or not, but farmers are not actually complaining about this resistance. Probably the herbicide selection which we have in Zimbabwe is not high enough to bring about weed population which is resistant to herbicides. You may find that the majority of smallholder farmers actually rely on mechanical methods, or they may combine mechanical methods and chemical methods, which is also common on large scale farming areas. The other point is that our seed banks, maybe they are very high. The seed bank size could be very high, and this can regulate weed seedling emergence. And few weed species with the genes of resistance could be present in the dominant populations, to the extent that they may not have a chance to reproduce. But there is need to determine the status of herbicide resistance in Zimbabwe. We need to do some studies. In conclusion, we can say that pesticide resistance in the pest organisms results from using the same pesticide repeatedly. Resistant weeds, insects, and fungi can limit the available management options for crop growers. Resistance can be managed in several ways so that pesticides remain a useful way of controlling pest organisms now and in the future. So this is it. So this is the end of pesticide resistance. Let me stop this.